First of all, I would like to thank the Department of Physiology for inviting me to contribute in this nice gathering. And I will tackle, in fact, the proteomics as a good tool that may contribute to better diagnose Alzheimer's disease very early in the course. Alzheimer's disease, in fact, the prevalence of Alzheimer's disease is increasing all the time, and as the population is getting more and more aged, we have more and more Alzheimer's disease among aged persons. And the risk factor to develop Alzheimer's disease as the most evidenced risk factor is still the age itself, rather than any other contributors. This is we will go through a quick reminder of what occurs in the brain in Alzheimer's disease. And we have here the normal brain on the left side and what occurs in Alzheimer's disease on the right side. And as you see that we have the amyloid plaques that are accumulated between neurons, and we have a neurofibrillary tangle inside the neurons. And to understand the amyloidogenesis pathway, we have to understand that the amyloid protein plaques is formed mainly of amyloid beta protein, which comes from a precursor, what we call APP, the amyloid precursor protein. And this is a transmembrane protein that plays a major role in contact between cells and the contact between cells and matrix. So to simplify, we have the amyloid precursor protein, which is the big molecule, and we have the fraction of the amyloid protein itself. And we have three series of enzyme cascade that breaks the amyloid precursor protein into different points to yield at the end the amyloid beta protein. So we have the alpha secretase, which in fact breaks the amyloid precursor protein at the middle of the amyloid protein itself. And then the beta secretase that in fact breaks the amyloid fraction towards the amino group terminal, and the gamma secretase that breaks the molecule towards the carboxylic outside terminal. So if I have the alpha secretase that breaks inside the amyloid protein, followed by beta, followed by gamma, I will not have non-soluble form of amyloid, which is the healthy pathway. On the other hand, if I'm going to skip the alpha secretase, I'm going to break the amyloid protein towards the amino group terminal by the beta secretase, and then the gamma secretase will move the other side, so I will pick this part, which is the fraction of the amyloid protein, apart, leading to amyloid protein. This is non-soluble form and it gets accumulated. And there are different types, in fact, or isoforms of amyloid beta protein according to number of residues that we have. So we have 37, 38, 40, 41, and 42. So we have two pathways. We have normal amyloid precursor protein breaking down cascade, and we have a beta amyloidogenic pathway that yields the non-soluble form of amyloid. And then comes the neurofibrillary tangles, and the neurofibrillary tangles are constituted of hyperphosphorylated tau protein. And tau protein, to understand simply, these are tubular-associated protein, and they are supporting the cytoskeleton of the cells. If they are getting more hyperphosphorylated, they will lose their function in supporting the cytoskeleton, and the cell will collapse. So if I have the tubular-associated protein, which is tau, so the unbound tau, or the tau will get unbounded from the cytoskeleton because of hyperphosphorylation, leading to aberrant assembly. Hyperphosphorylation takes place by different enzymes, but one of the major enzymes is the glycogen synthetase kinase. This is a very famous enzyme implicated in many psychiatric illnesses, and many researchers, by the way, are undergoing substances to inhibit this enzyme. So we have two processes. We have tau phosphorylation by phosphokinases and tau dephosphorylation by dephosphatases. And this balance is very important in Alzheimer's disease, and it's more towards tau phosphorylation. The CSF is in direct contact with brain parenchyma, and it can reflect many changes that occur in the parenchyma. And it's an easy way to have a sample of CSF with almost minimal adverse reactions. So To search for a good or ideal diagnostic test for Alzheimer's disease, these are some criteria that may be proposed by authors. It should detect fundamental features, it has to be validated by histopathology, precise, not invasive, simple, and not expensive. And so we will take what we can have in the CSF as measures to early diagnose Alzheimer's disease. Now it's well known that in Alzheimer's disease we have the total amyloid protein level that decreases, and it has been assumed that because of the amyloid is implicated in the plaques, so they are not free in the CSF, so the level is less, and the tau protein total is increased in the CSF. And those two features have been added, in fact, to the Alzheimer research diagnostic criteria. Most of the amyloid beta protein we have is amyloid beta protein 142. 42 designates number of protein residues that we have, and 1 because the beta secretase or the gamma secretase, in fact, can break the amyloid protein at certain protein, giving amyloid protein isoform 1 and isoform 2. We have sensitivity and specificity, and to be familiar with that, if I have a high sensitivity, it means that I have a very low chance to have a false negative result. So it's a good screening tool. And for specificity, means that if I have a high specificity diagnostic test, means that it can be used as a confirmatory test. So to measure the amyloid beta protein 42, we have a specificity and sensitivity around 80%. However, we can improve that. We can improve that through rather analysis of the different isoforms of amyloid beta protein rather than measuring the amyloid protein in its totality. So we have amyloid beta protein 142 that is decreased, and this is the most studied one, but we have other isoforms as well that are less studied, like amyloid beta protein 2, 42 residuals, and we have different subtypes like the 42, 40, 38, and all of them are increased, while amyloid beta protein 142, which is most studied, is the only one that is decreased. So what's important, in fact, is not to study the amyloid protein, the total amount, but to study the different ratios between different isoforms that may enhance the specificity and sensitivity of this, as we are going to mention later. And then comes the phosphorylated tau, and the phosphorylated tau level, in fact, increases with age, and it's very high in the fetal period, and then decreases a little bit during adult period, and then increases once again in Alzheimer's disease. So that's why we have the phosphorylated tau level that should be adjusted according to the age. So the quantity of neurofibrillary tangles for which the phosphorylated tau protein are mainly incriminated is in fact more sensitive to the staging of Alzheimer's disease rather than amyloid plaques. Many disorders have amyloid plaques and patients in postmortem, but in fact they did not present any Alzheimer's disease. So we have different phosphorylation sites, and these are sites that are very interesting. We have tau protein that's phosphorylated at the site 153, 262, and 231, and these are more present in the early course of Alzheimer's disease, while we have others like 199, 202, and 205, and all that figures out in the right square, and those are increasing in the late Alzheimer's disease course. And these are the different enzymes that are involved, particularly the glycogen synthetase. So we have 79 points where phosphorylation of tau protein can take place. So the mean sensitivity and specificity of phosphorylated at different position varies from 44 to 94%, and specificity between 80 and 100%. So let's take a look. If we are measuring the total tau protein in the CSF, I have 20% chance to get a false negative result because my sensitivity doesn't exceed 80%. But if I'm rather measuring the phosphorylated tau protein at the site 396 or 404 ratio to the total tau protein, I increase this sensitivity up to 69% and the specificity up to 94%. which is a very important from a clinical perspective, to have only 4% false negative and only 6% false positive. So, analyzing the phosphorylated tau protein at the level or at the amino acid number 396 or 404 are more informative than simply measuring the total tau. So, tau protein is in a dynamic process of the time. It's a continuously dynamic process between phosphorylation and dephosphorylation. And the dephosphorylation of the hyperphosphorylated tau is one of the main focus under research to try, in fact, to reverse once again the changes that occur in Alzheimer's disease. Another interesting thing, phosphorylated tau at the site 199. This can discriminate Alzheimer's disease from other groups of aged persons with a sensitivity of 85%, which is 15% higher than the total protein tau in total. Another interesting phosphorylated tau biomarker is the phosphorylated tau at the site 231. And this particular isoform has another behavior during the whole course. It increases gradually and then decreases as the Alzheimer's disease course is advancing. And this means that it's an excellent biomarker to trace, in fact, the advancement or the progress of the illness. Another interesting isomer is the phosphorylated tau at site 231 and 235, because they can differentiate with a high sensitivity aged persons with minimal cognitive impairment who are going to develop Alzheimer's disease. And this is the main challenging point, in fact, from a clinical perspective, because even the amyloid immunotherapy, for instance, once the pathology has been initiated, they are not effective at all. So the challenge is that to pick up patients who are going to develop Alzheimer's disease rather than to try to treat Alzheimer's disease patients themselves. So many biomarkers are suggested by authors: the tau, MAO-B, apolipoprotein, amyloid beta protein, P97, and the NTP, which is also agreed. This is the neuronal thread protein. Another era is coming is that if I'm not only going to rely on the tau protein or the amyloid protein, but I'm going to combine both together. So this may enhance much better the sensitivity and specificity of tests. So if I'm going to combine, for instance, the tau protein and the amyloid beta protein, in 40. So if I'm going to measure tau only, I will have a sensitivity around 80%. If I'm going to measure the amyloid beta protein depleted sheet 42 residues, I have a sensitivity of 74. But if I'm combining together both measures, I will have a sensitivity up to 86%. This means if I have a patient who has the tau protein that is increased and amyloid beta protein that is decreased, I have a sensitivity of 86 rather than 80% if one of them has been done only. Another thing, if I'm going to measure the total tau protein in the CSF, I will find it increased. And if I'm going to measure the ratio between the amyloid beta protein 42 to the amyloid protein 40 rather than to measure the total amyloid beta protein, I can increase the sensitivity up to 94%. And those figures are really very interesting in terms of clinical application. So the proteomic analysis, and I think Professor Passant has already given a lecture on the proteomic techniques, is really giving a great hope to better refine our diagnostic test to understand what we can do. So it's not important to measure the protein itself, but to try to go more through deep analysis of the different isoforms of a single protein, because it will be much more informative. And finally, to conclude, proteomic analysis may really contribute largely to improve diagnostic tests, and it can contribute not only for the diagnosis, but to trace the cause and to improve the therapeutic outcome, and even to predict outcome from certain medications. And finally, which is very challenging from a clinical point of view, is to pick up patients with minimal cognitive impairment that has been nomenclatured in the past normal aging. Who are among those persons, or what we call normal aging, that are going to develop Alzheimer's disease out? Thank you very much.